afternoon, chairperson, eminent scientists, and my colleagues. Here I am presenting my paper entitled Reversal of Acrylamide-Induced Toxicity by Therapy of Kaempferol in Rats. Acrylamide is known as group 2A carcinogen. It is a neurotoxic in human, reproductive and developmental toxicant in animals. This chemical produced by the carbohydrate food is cooked at high temperature due to Maillard reaction. And this average daily dietary intake of children is 1 microgram per kg and adult 0.3 to 2 microgram per kg. And these foods have the 500 microgram per kg label, especially French fries, wafers, biscuits, bread crepes, coffee, and etc. These pastries, bread rolls, potato chips, and coffees we are mandatory in our breakfast. If we can fry French fries at the temperature of 185 degrees Celsius for 5 minutes, its level reaches 1500 microgram per kg. Acrylamide ingestion caused ataxia, vertigo, pupil reactive to light, and it caused the organ failure. So McDonald's and Burger King agreed to provide cancer hazard warning regarding acrylamide in their French fries due to case filed by the CRT California. Acrylamide is exposed by these 3 routes: oral, transdermal, and inhalation. And this acrylamide ingested by the human being, and it metabolized in the presence of cytochrome P450 into glyceramide and goes to our body in the form of acrylamide and glyceramide. This acrylamide causes the effect on nervous system, reproductive system, and it caused the cancer, and it also disturbed the endocrine system. So human exposure to acrylamide of great concern. Therefore, research on therapeutics is needed well before any alarm of danger. So my aim of study is impact of polyphenolic compound kaempferol against acrylamide toxicity. This aim will be achieved by following objectives: whether plasma membrane damage and glutathione homeostasis, measurement of liver, kidney and brain function test, influence on DNA damage, reversible inhibition of cytochrome P450, and histopathological and ultrastructural studies. Kaempferol is a natural flavanol, strong antioxidant and chemopreventive agent for cancer. This is present in tea, broccoli, grapefruits, beans, apples, and it has the properties like as antioxidant, anti-inflammatory, anti-allergic, hypolipidemic, and vasoprotective, and anti-carcinogenic, anti-hypertensive, and anti-thyroidogenic. This is our protocol 1, which deals with the selection of the optimum dose of kaempferol. So we have selected animals, our departmental animal house, and randomly divided into 6 groups, 6 animals each. Group 1 served as a normal control, group 2 is kaempferol per se, and group 3 is administered at a dose of 1/3 of LD50 of acrylamide for 10 days. And therapy will be given of group 4 to 6 at different doses of kaempferol. And following serological function test and tissue parameter will be done. This is acrylamide cause elevation in the lipid peroxidation in liver, kidney, and brain, whereas it decreases the level of reduced glutathione in all the organs. Therapy with kaempferol at all the doses showed recovery pattern. Maximum recovery was shown by both the higher doses. The activity of superoxide dismutase and catalase were decreased by the exposure of acrylamide. Therapy will be restored all the parameters in all the organs, and maximum recovery was found with both the higher doses. Acrylamide also disturb the brain function, kidney function test, which is clearly shown by the elevation in the urea and creatinine, and it is also increased the level of lipid profile, and therapy with the kaempferol showed recovery pattern. So the outcome of this protocol is both the higher doses of 20 mg per kg and 40 mg per kg was found to be effective in preventing acrylamide toxicity. Next, so this is next protocol which we done against the subchronic exposure, which confirm the kaempferol is protective against the acrylamide. Here we take the animals and we randomly divide it into 3 groups. Group 1 is served as a control, and group 2 is administered at acrylamide at a dose of 1/6 of the LD50 for 28 days, and group 3 is treated with the kaempferol at the dose of 20 mg per kg. And following blood parameters, brain function test, tissue parameters, drug metabolizing enzymes and DNA damage were performed. Acrylamide decreased the level of AST, ALT and alpha-levulinic acid dehydrogenase, where it increased the level of serum alkaline phosphatase and urea, and the therapy with the kaempferol showed the recovery in all the parameters. Acrylamide is known as neurotoxicant, which is clearly shown by the inhibition in the activity of acetylcholinesterase in all the parts of brain. And the therapy with kaempferol showed restoration. And acrylamide caused the elevation in total cholesterol, esterified cholesterol, triglycerides and alpha-levulinic acid synthetase in brain. And the kaempferol showed restoration in all the activities. This is an antioxidant enzymes. They are also decreased by acrylamide exposure, glutathione reductase, glutathione peroxidase and glutathione S-transferase, where the activity of glucose 6-phosphate dehydrogenase is increased. Therapy with all kaempferol showed recovery in all the parameters, but it recovered very less in the glutathione peroxidase in liver and kidney. Acrylamide also decreased the level of aniline hydroxylase and amidopyrine methylase, which is clearly shown it damaged the liver also, and whereas it increased the level of cytochrome P450 and microsomal lipid peroxidation. Therapy with kaempferol showed recovery in all the parameters, and acrylamide convert into glycidamide in the presence of cytochrome P450, which is form the DNA adduct and cause DNA damage, which is clearly shown by this tail length of the hepatocytes and DNA damage, and it also clearly shown in the kidney cells and it also shown in the brain cells. Recovery is shown by the kaempferol in liver cells and kidney and brain cells, and acrylamide cause damage in the hepatocytes, which is clearly shown by the vacuolation and hypertrophy in hepatocytes. It caused the hypertrophy in glomeruli and degenerated endothelial lining, and it caused the hypertrophy in epithelial cell of the renal tubules. It also caused the damage in the pyramidal cells of the brain, where the therapy with kaempferol at a dose of 20 mg per kg showed recovery in all the organs. This is photomicrograph of normal control, which is shown the terminal buttons, well-formed mitochondria, synapse, synaptic vesicles, and myelin sheath. And whereas the acrylamide cause demethylation in the axons, and it is also cause damage in the mitochondria, and it regenerate the cristae of the mitochondria. It also disturb the presynaptic membrane and postsynaptic membrane, and it, whereas the therapy with kaempferol should maintain mitochondria and well-formed myelin sheath of axons. Acrylamide is readily absorbed by gastrointestinal tract and metabolized into glycidamide. Some amount of acrylamide and glycidamide form the conjugate with the GSH and form mercapturates and excrete out from the body. Some amount of acrylamide and glycidamide form the hemoglobin adduct and cause oxidative stress in blood biochemistry, tissue biochemistry, and histopathological observation. Some amount of glycidamide form the DNA adduct and cause the DNA damage, which is the cause carcinogenesis. And this kaempferol is helped to maintain antioxidant status, anti-apoptotic, and it is neutralize the reaction oxygen species and regulate inflammation. So thus, we concluded this present study, kaempferol will be a novel therapeutic agent against acrylamide toxicity. Here I am acknowledged and thankful to our financial assistant agency, Department of Science and Technology, New Delhi, Jivaji University, for providing me laboratory facility and this conference to providing to share my work with you. Thank you.
This talk is open for discussion.
Please.
Yes, sir.
The number of animals, 6 for each group, yes?
Yes, sir.
It is statistically non-valid. This number is very small.
Yes, sir.
Number one. Number two, you measured total cholesterol.
Yes, sir.
Medically, it is insignificant. You have to measure HDL and LDL cholesterol.
Okay.
Total cholesterol means nothing.
Yes, sir. Next experiment, I did this HDL and LDL also.
Yes.
But now I am only measure the total cholesterol and extracellular cholesterol in the brain tissue. And afterward, I can do it in chronic exposure.
You measured lipid peroxidation, yes?
Yes, sir.
What's the parameter to indicate lipid peroxidation? Huh?
TBRS.
What?
TBRS. Through the TBRS, thiobaritic acid.
The most important is protein diols for lipid peroxidation. Protein diol or MDA.
Not MDA, by the TBARS.
Okay, thank you.
Thiobaric acid.
Thank you.
Any more questions? Oh.
Namaste, Dhanyavad, for your excellent presentation. My question is, which industries or which geographical locations do you find the most acryl amide in human beings in your country?
Yes, ma'am. Please, question again, please.
Sorry. In your country, India,
Yeah.
which people, which occupational groups or which industries have the people most affected by acryl amide as a carcinogen?
I'm not a steady human being, but this is exposed by, it is present in all the carbohydrate food, which is, we can fry at high temperature. So acrylamide is formed due to the Maillard reaction. So it's present in all the foods, almost kurkure, chips.
She's asking, eaten by which group most? Maybe children, no?
Young ones, children's.